Hello everyone. Welcome back. Today we're going to be discussing chapter 3, which is entitled Cells. Now, personally, this is one of my favorite chapters because it explores the cell theory and it allows us to talk a little bit more about the differences between a prokaryotic and a eukaryotic cell, as well as starting to take a look at organelles such as like the nucleus, the mitochondria, the smooth and the rough endoplasmic reticulum, and how they collectively work together to contribute to the overall well-being and function of the cell. But before we get to all those details, let's talk about this thing called the cell theory. Now, in chapter 1, I had discussed with you the fact that part of the scientific method is to allow for hypotheses to be transitioned into a theory. So that is based on the fact that scientists will collect more and more research about a phenomenon and they'll get more knowledgeable and better at understanding it. And eventually it will become a theory. One of those topics that a lot of research has been done on are cells. So we have the cell theory. And the cell theory tells us three things. It has what they call three tenets. The first thing is that all organisms are made of at least one cell. So we're looking at eubacteria, which are unicellular form, but then also we're looking at plants, at animals, and fungus, at protista. Those are multicellular organisms. The second part is that the cell is the fundamental unit of life. So everything starts at the cell surface. And the third one came about a little bit later, and it's partly due to the discovery of the microscope. And it says that all cells come from pre-existing cells. Now, this one has a little bit of a backstory behind it because before we knew of these microscopic gametes and things that we couldn't see without the help of a microscope, scientists had this thing called spontaneous generation. And spontaneous generation basically stated that if you have an inanimate object, so something that's not alive, if you put it in the right environment, surrounded by the right conditions, it would spontaneously generate life. So for instance, if you have some meat and you leave it out in the hot sun, spontaneously maggots would arise from it. Or better yet, if you live next to a water source, like maybe a lake, come summertime when it gets nice and hot, spontaneously tadpoles and frogs would be generated. And I know it sounds very silly, and we know that these things don't spontaneously generate, but this is just an example of how scientists didn't have the knowledge because they didn't have the right tools. Once the microscope made an appearance, we were able to see microscopic items such as the different gametes, and we were able to explain things like spores, and then understand that these cells did come from pre-existing cells, not inanimate objects. As we peruse through the chapter, we're going to talk a lot about differences between cells. So I think it's important to also point out what all cells have in common. And on this slide right here, we're seeing 6 items that we're going to have in any type of cell we look at, whether it is a simplistic cell like a prokaryotic cell, or it is a more complex cell like our eukaryotic cell. Now, the first 4 items are kind of bundled together, and it basically states that all cells will have DNA because that is their genetic information. And based on their DNA, cells will be able to create RNA. And once they have RNA, they can utilize ribosomes to create proteins. So DNA gives you your genetic information. This is your adenine, thymine, guanine, and cytosine working together in the form of your chromosomes. And then DNA is able to transcribe or produce RNA, ribose nucleic acid, and RNA will then carry the message of DNA, but more importantly, will give all the information to produce proteins. And in order to produce the proteins, we do require ribosomes along the way. Now, from a structural aspect, all cells will have a cell border. This will be their plasma membrane. This will be almost like their border that will separate the inside and the outside of the cell. And on the inside of a cell, all cells will have a cytoplasm. The cytoplasm is the general area inside of a cell. It is filled with this fluid called cytosol. And within this fluid, within this area, you will then have your DNA And if it's a eukaryotic cell, you will have your organelles that are floating around. So please keep that in mind. Regardless of what type of cell you look at, you will always have DNA, RNA, ribosomes, proteins, and then from a structural aspect, you will always need a cell membrane, also called a plasma membrane, and the area inside of it is called the cytoplasm. In a little bit, I'll show you some illustrations of a labeled cell so we can kind of take a better look at that plasma membrane and that cytoplasmic area. But before we do that, I want to talk about another feature that we have in cells. Cells all typically tend to stay nice and small, and part of that has to do with their surface area to volume ratio. And what these items tell us is based on how much nutrition and waste product a cell will generate and the ways that they're able to go ahead and get the nutrients in and the waste products out. So let me go ahead and show you an example where we look, for instance, over here, and we can look at the size of a cube. Excuse me. And as you can see, we have 3 cubes, starting with the smallest, 1 centimeter on each side. And then we're going to start doubling it to 2 centimeters. And in our third scenario, we have one that has been tripled to 3 centimeters. Now, all of them are relatively small, but obviously the 1 centimeter is smaller than the 3 centimeters. And what we're going to do next is we're going to go ahead and calculate the surface area. Now, the surface area will be basically what's happening on the outside of the cell. So we're going to multiply the height times the width times the number of sides. And what the surface area allows us to see is how much opportunity or how much area does the cell have for things to come in and out. The second thing we're going to calculate is the volume. Now, the volume is what's happening inside the cell. So that's going to be the height times the width times the length. And the volume, you could almost think of it like your cytoplasm. What is happening on the inside of the cell? This will determine how much nutrients the cell needs and how many waste products it's going to generate. Okay, so surface area is what's happening on the outside. So in order like your plasma membrane, it's going to be the method that you get stuff in and out. And then the volume is what's happening on the inside of the cell, like your cytoplasm, and it's going to determine your nutrition and your waste products. Now, as you can see, for each of the 3 cubes, the numbers have been calculated. And as we can predict, as the cube gets larger, the surface area gets larger and the volume gets larger. However, they do not enlarge or increase at the same rate. So for instance, if I go from 1 centimeter to 2 centimeter, I see that the surface area will increase by a 4-fold. It will go from 6 to 24, but the volume will increase by an 8-fold. And that trend will tend to continue. The volume, the inside of the cell, continuously increases at a much faster rate than the surface area, the outside of the cell. And we can also see that by the bottom of our illustration where we're looking at the ratio. So the ratio is basically comparing surface area to volume, and it's coming up with a numerical value. And what we start seeing is as the cell gets larger, this numerical value starts to decrease. And decreasing is a bad thing because it means that the cell will not be able to keep up with its nutritional and waste demands because its volume will increase dramatically faster than the outside, the surface area, which is how we're going to get stuff in and out. So in an ideal world, you would want a cell that's nice and small because it will give us a high ratio, which means plenty of opportunity to get stuff in and out through the surface area and keep up with the demands of the volume. As the cells get larger, this ratio decreases, which is not good for the cell. Now, there are a few exceptions to the rules. One of them is that if you need a cell that needs to be relatively large, well, we can make it long and flat, which is the case of what we see, for instance, in our neurons. Or we can take a look at our amoeba. The amoeba has these things called pseudopods, which means it can extend its cell membrane and make it very fluid, allowing for the cell to be relatively flat and also increase its surface area by its stretching movement. Very cool. Now, speaking of our prokaryotic cells, remember, just as we discussed in chapter 1, these are going to be the cells that lack the nucleus. So they still have their DNA, they still have RNA, ribosomes, and proteins, and yes, they will still have their cell membrane and the cytoplasm, but the DNA will be freely floating around. Eukaryotic cells, on the other hand, are the ones that do have a nucleus. And in a little bit, we're going to talk about the fact that since they do have a nucleus, they will be able to be slightly larger and definitely more complex than the prokaryotic cells. At the bottom of the slide, we see the reminder of our 3 domains we've mentioned before. The bacteria and the archaea are both classified as prokaryotic, and then the eukarya domain gets its name because it is a eukaryotic cell domain. Here is a nice little figure from your book, figure 3.5. I really like this because it compares the 3 domains and it reminds you of what the cell type is like. It tells you what's happening with the nucleus, and it also mentions things like the cell wall. Now, the cell wall is an additional protection. So imagine that this is your very simplistic cell. This represents my cell membrane. Some organisms have an additional layer on top of this, and that layer is called the cell wall. Animals, such as ourselves, we do not have a cell wall, but you can find the cell wall very commonly in bacteria, in archaea, as well as in fungi and in plant cells. Now, let's talk a little bit about the domain bacteria and archaea, and then we're going to switch our focus over to our eukaryotic cell. For the domain bacteria, I want to remind yourself that based on our previous conversation, we are looking at a unicellular organism, so an organism that's composed of one cell. Very simplistic in its structure. It does not have a nucleus, but it does have what's called a nucleoid. And a nucleoid is a very simplistic structure. I like to think of it kind of like a little envelope that will freely house the DNA. It will also often have a cell wall, which is that additional structure beyond the plasma membrane. And the cell wall is very beneficial for structure as well as for protection. Bacteria usually come in 3 shapes. We have what we call the rod shape or bacillus. We have the circles or the round, which is called cocci, and we have the spiral ones or spirillum. I will show you an illustration of this in 1 second. Often bacteria will also use this concept of a flagella, which looks like a whip-like structure. It looks like a little tail. It will use it for movement. Here you go. Here is a very simplistic structure of a prokaryotic cell, more specifically a bacteria cell. And as you can see right here in blue, this is the cytoplasmic area. Here we see the DNA floating around. And then these little green dots, those are the ribosomes. Remember, all cells have ribosomes. If we now take a look at the borders of the cell, we see that we have a yellow, a purple, and a green. The yellow is going to represent the cell membrane, which all cells have. The purple will represent the cell wall, which is that additional layer of protection. And this bacteria has a 3rd layer. It has a capsule layer. Not all bacteria have capsules, but the capsule layer is usually composed of a sugar component, which allows it to freely stick to different surfaces, which of course is very beneficial because it allows them to interact better with their environment. These tail-like structures, that is the flagellas that I was just talking about. And here's a cool little fact. As humans, we only have 1 cell that has a flagella, and that is the sperm cell. Below, we see our 3 shapes. This is the bacillus or rod shape. Here is the cocci or circular or round shape. And here is the spirillum or spiral shape of the bacteria. Aren't those beautiful? Now, the next domain is the domain archaea. And for the domain archaea, we're going to keep it a little bit short because we will be talking about it more in future chapters. For now, I just want to remind you that it's also prokaryotic. It is also unicellular. And to be very honest with you, the domain archaea, its organisms have a lot more in common with eukaryotic cells than prokaryotic cells when we take a look at their structural components. But remember, they don't have a nucleus. So that is then why they are classified as a prokaryotic cell. And a lot of the archaea are known as, or I shouldn't say a lot, my apologies. Some of them are classified as what we call extremophils. And an extremophile means that they like extreme environments, such as extreme heat, very acidic. In fact, if we look at our little illustration right here, we see Solvulus, and this unicellular organism loves to live in a hot spring with 80 degrees Celsius. Remember, our internal body temperature is 37 degrees Celsius, so this is quite hot, and a pH of 2, which, as we discussed in our previous chapter, is going to be extremely acidic. Now, I will also let you know that you can find archaea in regular environments. There have been studies that have shown that, for instance, within our mouth, we not only have bacteria, but we also have some archaea that can make a home. So that's why it's so important to keep brushing those teeth, right? And flossing. This then leads us to the domain Eukarya. Now, the domain Eukarya will have a wide variety of different organisms that we're going to explore here. And some of them can be found in the fungi kingdom, so like your mushrooms, the plants, the animals, and then the miscellaneous category of the protista. What I would like to do is focus mainly right now on plants and on animals. And then along the way, we're going to pick up more knowledge about the fungi and the protista. Now, a eukaryotic cell does tend to be larger than a prokaryotic cell, which I know sounds a little bit odd because we just discussed how the cell likes to stay nice and small. But it turns out that eukaryotic cells, because they will have a nucleus and other types of organelles, they are able to divide up the labor within the cell and still be energy efficient. So that is one of the reasons why they can get away with being slightly larger than a prokaryotic cell. Now, what I want to do now is I want to show you a picture of an animal cell, a typical animal cell, and then a typical plant cell. And we're going to take a look at some of the things that plants have, but animals do not. So here is my typical animal cell. As you can see, a beautiful nucleus, which is what makes it a eukaryotic cell. These little blue lines that you see, it always looks like a little tie-dye to me. Those are your chromosomes or chromatin. And as you can see, it's being housed in a cell that has a cell membrane. Here's the cell membrane in the yellow color. And all the area right here, this is our cytoplasm. And you can see the different organelles floating around, like our mitochondria, our Golgi apparatus, our rough and our smooth ER. We're going to talk about what these contribute to the system in just one sec. Now, here's your animal, and here is your plant cell. Once again, beautiful nucleus. And now we see two things. We see that it has a cell membrane, just like we saw in the animals, but on top of it, it has an extra layer. That's right, plants have a cell wall, just like we saw in our bacteria sample that we were just looking at. This cell wall is made out of cellulose, and this is one of the features that a plant cell has that an animal cell does not. A second item that a plant cell has that animals do not have is this large structure right here. It's called the central vacuole, and what the plant will use this for is to store its water supply. And as it stores water, the vacuole will increase in overall size, creating turgor pressure, which is partly what the plant used to stay nice and erect. The third organelle that plants have that humans do not have, or I should say animals do not have, is this green item right here. You can find one here. Here's the second one. Here's the third one. And this, ladies and gentlemen, is chloroplast. Plants rely on chloroplast for their photosynthesis because, as you may recall, they are autotrophs, so they're able to generate their own food. We are not autotrophs, we are heterotrophs, and we do not photosynthesize. So that's why our cells will not have any chloroplast. So just to kind of reiterate, three things that a plant cell has that a animal cell does not have will be a cell wall for additional protection made out of cellulose, a central vacuole for water storage, and chloroplast for photosynthesis. The cell membrane of the eukaryotic cell, whether we're looking at an animal cell or a plant cell, is composed of what we call phospholipids. And phospholipids are going to be a combination of glycerol, fatty acids, and a phosphate group. And it turns out that these three items will interact, create what we call a head region. So here's my little ball. This represents my head region, and a tail region. Another important point to point out is that the glycerol and the phosphates, which were located in the head section, will allow for a hydrophilic region to be generated. So remember, hydrophilic means water-loving. The tail, on the other hand, since it's composed of fatty acids only, of lipids, that will be hydrophobic, water-fearing. And to me, that's very interesting because we know that our body is made out of a lot of water. There's water inside the cell, there's water outside of the cell. So why would we want to have a cell membrane that is part water-loving, hydrophilic, and part hydrophobic? It turns out there is a good explanation for that. But before I tell you the explanation, I want you to see how it's actually set up. So first off, the cell membrane is what we call a bilayer. So it will have two layers of these phospholipids. Here's the first one, and here's the second one. And notice how the orientation of the phospholipid bilayer is that the heads, the parts that are hydrophilic, are pointing outwards, meaning that they are exposed to the outside of the cell and they're exposed to the inside of the cell, the cytoplasm. That's the area where all the water can be found. The hydrophobic tails are folded inwards, so they are shielded from the water component. Very smart. And it turns out this format and the use of a hydrophilic head and hydrophobic tail allows the membrane to become selectively permeable. And selectively permeable discusses the fact that the cell membrane will be able to control what comes into the cell and what goes out of the cell. So for instance, you want to make sure that your nutrient can come in, your waste products come out, but you want to make sure also that your organelles or your DNA stays inside the cell and doesn't freely float away. So this is one of the reasons why it requires a hydrophilic and a hydrophobic component, a water-loving and a water-fearing component, all in the name to become selectively permeable. I also want to point out that often when you read about the cell membrane, you'll see a description said fluid mosaic model. Fluid because the membrane can easily change shape. It is not a rigid model. It can change its overall shape in the environment that it is. And mosaic means that it's made out of different components. The main components will be the phospholipid bilayer, but embedded between that, you will also find different proteins that have different functions. So here is this beautiful picture of the cell membrane. Starting on the left-hand side right here, we see the animal cell membrane. So we see our little phospholipid bilayer with the heads and the tail. And if we look ever so closely, we also see some cholesterol embedded. Cholesterol will actually allow for the fluidity of the cell membrane to be manipulated. And then these large purple items, these are different proteins that are embedded in the cell membrane. And they'll have different functions like transport, communication, cell identification. If we now compare the animal to the plant cell, we notice a very similar setup. We have our phospholipids, we have our proteins, but then we also have this large green structure that's set up on top of it. And that's just a quick reminder that your plant cells have a cell wall for that additional layer of protection. Now, let's get a look at what's happening inside of the eukaryotic cell itself. And inside of it, we have this thing called an endomembrane system, which means that you have little organelles that have specific functions. And they will all work together, and everything will start with our DNA. Because remember, our DNA is our genetic code. And in eukaryotic cells, the DNA will be housed in the nucleus. So what I would like to do is I would like to show you how we go from having DNA to actually communicating with the cell and being able to produce a lot of different secretions, a lot of different proteins, a lot of different hormones. And in the example of the book, it talks about making milk. So here's our beautiful little illustration where we can see we have our little piglet and it's suckling on its mom because it's looking for milk. So the question is, how does the cells from the mom know that it needs to produce milk, and how will it produce the milk? Well, in order to explain that, we're going to take a look at this illustration right here, and we're going to take a look at how the different organelles will play a role in producing milk for the piglet to enjoy. So we're going to come back to this picture because I want to show you what the name of these individual organelles are, and then we're going to talk about how they contribute, and then we're going to put it all together at the end. So let's start off where we need to start off with, which is we need some information. We need our genetic code. So for that, we're going to go over to the nucleus. The nucleus is where we house our DNA. And if you see terms like chromatin, chromatids, chromosomes, genes, all we're doing is we're just giving a different name to our DNA, our genetic information. Think of your DNA almost like a cookbook. It has all the recipes that you need. So if your body or your cell needs anything, it always needs to go over to the DNA and it needs to get its set of instructions. The DNA is housed in the nucleus for our cells because we are eukaryotic organisms, and the DNA cannot leave the nucleus. So instead, what's going to happen is that the DNA will make a copy of its directions in the form of RNA. And RNA is able to go in and out of the nucleus as it pleases. So DNA stays inside, but RNA can leave, and it will use these little openings called nuclear pores that are all over the nuclear envelope of the nucleus. Inside of the nucleus, you also have another entity. This is called the nucleolus, which literally means mini nucleus. This one is responsible for producing ribosomes. Now, we've seen the term ribosomes before. Those are some of the things that all cells have in common because they're going to help us produce proteins, just like we need the DNA and the RNA to get that done. Here is an illustration of your nucleus. So let's start on top. Here we have our animal cell, and as you can see, they've highlighted for you the nucleus area. You can see that it has a nuclear envelope that makes up its border with little openings in it called nuclear pores. And the DNA is indicated by these little tie-dye streams. Keep in mind the DNA has all the directions, but it cannot leave the nucleus. So the DNA will form RNA, which can then use the nuclear pores to go in and out. And right over here, we have our nucleolus. That is what we're going to rely on to produce our ribosomes. Okay, so in our example, we see that we have a mama who needs to produce milk. So the first thing our cells are going to do is they're going to go within the nucleus and get the directions from the DNA, which will make RNA. And the RNA will then travel out of the nucleus into the cytoplasm and will start interacting with the different organelles that are located there. Now, for our case, the first organelle we're going to talk about is one that is very close to the nucleus. As you can see right here, it's surrounding it. It's going to be called the endoplasmic reticulum, or for short, ER. And the ER comes in two variations. We have the rough ER and we have the smooth ER. The rough ER gets its name because when we look at it, we see that it has these ribosomes, here shown in green, studded all over the place. So it makes it rough in its appearance. The smooth ER does not have ribosomes. It just has a smooth surface. The rough ER, because of the use of the ribosomes, is where we're going to be able to produce our proteins. And the smooth ER is going to help us produce lipids or fats. So think of these as individual ingredients, right? So you want to make something, you have your directions. So now we're going to go over to the grocery store and we're going to get our individual pieces. So we're going to go into one aisle for the rough ER and get our proteins. We're going to go into another aisle for the smooth ER and get our lipids or our fats. And also, by the way, the smooth ER does many other things, including producing enzymes that help us detoxify drugs and poisons that might be introduced into our system. In fact, the organelles that we discussed today, all of them have multiple functions, but I'm just kind of highlighting the most important ones, and of course, the ones that are relevant to our class. So now that you have your proteins, thank you, rough ER, and now that you have your lipids, thank you, smooth ER, let's go ahead and figure out how to put all these ingredients together to get to our final product, which in our example is the production of milk. For that, we're going to have to go to the Golgi apparatus. Some books will reference this as the Golgi complex. This is going to be the organelle that's going to take the individual pieces like the proteins and the lipids. It's going to sort it, it's going to package it, and it's going to be able to produce your final product. It doesn't matter if it's going to be milk or a particular hormone or a particular enzyme. The Golgi apparatus is almost like the post office, FedEx, like UPS. It puts everything together and it gives you your final product. The Golgi apparatus tends to be located towards the edge of the cell, and the way products come in and out is through the use of what they call vesicles, which are small containers. These vesicles will enter the Golgi apparatus, which looks like a series of flattened sacks. It will enter the Golgi apparatus, it will be sorted, it will be packaged, and once you have your final product, there are other vesicles on the shipping side that will allow it to exit the Golgi apparatus. From there on, the product can stay inside of the cell, or in the case of the milk product, it will be excreted through the milk ducts so that the piglet can enjoy its next meal. So here is that picture again that we started our conversation with. And if you don't mind, let's take a look at all these different organelles now at work, now that we hopefully have a better understanding of it. So here in blue is our nucleus, and it shows us that the DNA is being converted into RNA because RNA can leave, DNA cannot. Once you have your directions, you will go over to the rough ER. See the little ribosomes studded all over the place for your proteins. You will go over to the smooth ER for your lipids, and then your little vesicle, your little container of individual ingredients will enter into the Golgi apparatus where it will be sorted, where it will be packaged, and your final product will be then placed in a little transport vesicle. And in our example, you can see it will exit the cell and it will travel over to the milk duct so that you can have a happy baby that has some milk. And of course, if milk is not relatable to you, keep in mind that you're going to do the same exact steps, but you're going to have different products. But it will always start with the nucleus because that is where we house our DNA, our genetic information. Now, in case you're wondering, are there other organelles out there? Yes, there are a boatload of different organelles that are out there in the cell. Some of them are used for maintenance and what they call housekeeping. One of them is, for instance, the lysosomes. Lysosomes get their name because they do hydrolysis. If that term seems familiar to you, it's because we discussed it in our previous chapter. It's when you use water to lyse or to pull items apart. In this case, lysosomes will use hydrolysis to destroy any type of damaged organelle or perhaps some debris that has entered the cell that we want to get rid of. The lysosomes is able to do that cellular digestion. We also have peroxisomes. Peroxisomes tend to focus more on toxic substances. They love, well, I shouldn't say they love, but they primarily focus on what they call blood toxins. So you will find them in cells that are like the liver and the kidney cell that have high exposure to blood and therefore would also be able to sense these blood toxins. Peroxisomes get their name because they produce something very similar to hydrogen peroxide as a way to dispose of these toxin substances. Oh, I see the vacuole is also listed on this list. Now keep in mind the vacuole, that's for the plant cells. We don't have that. And part of that is because the plant is going to use it for the water storage, like we were chit-chatting about, but also because plants don't have lysosomes. So they will use the vacuole also for that particular cellular digestion function. Here is a look at some peroxisomes. As you can see, they have little protein crystals inside that they're able to generate and get rid of the blood toxins. And it's quite common to have several peroxisomes within the cell. Of course, depending on if you're looking at a cell that has a high exposure of the blood toxins, they will have more peroxisomes than others. Another organelle that is actually one of my favorites, and I think a lot of you have probably heard of it before, the mitochondria. The mitochondria is the powerhouse of the cell, and that is because it allows us to take the energy that we have in the nutrients that we consume, and it allows us to form ATP, which stands for adenosine triphosphate. And think of ATP almost like your battery charge. It is a form of energy that your body uses to get a lot of the work done that needs to get done. And it is all produced by the mitochondria. Now, the mitochondria has a very interesting story. Part of it is that when you look at the structure of the mitochondria, what you're going to notice is that the organelle, and here you have it, has what we call a double membrane. It has a membrane on the inside that is full of curves, and it's fully functioning and it helps us with the ATP production. And then you have a membrane on the outside, which is nice and smooth and has no function at all. It's almost as if it's there just to say, hey, I belong to the cell and this is the border that was placed above it, right? And it turns out that there's some truth behind this because based on scientific research, the mitochondria is an example of an organelle that came to our cell due to the endosymbiosis theory. And the endosymbiosis theory tells us that millions of years ago, the mitochondria was a free living cell. So it was its own cell, its own organism. It was engulfed or eaten by a larger cell. And instead of being consumed, the larger cell made the mitochondria part of its genome. So the mitochondria then became a part of the eukaryotic cell. And I know it sounds a little weird. It kind of reminds me of Pac-Man, where you have, the large Pac-Man eating the smaller ones. But there's evidence because it's a theory. The evidence is that one, mitochondria has that double membrane. So the inner membrane belonged to the original cell, and then the outer membrane was placed there by the host cell. And the second piece of evidence is that mitochondria has its own DNA. All the other organelles that we've talked about, the smooth ER, the rough ER, the Golgi apparatus, they rely on the instructions from the DNA in the nucleus to tell them what to do. Mitochondria has its own DNA. It's called mitochondrial DNA. So that means that in essence, it can perform its functions all on its own. In reality, it relies on the DNA from the nucleus to get most of its work done. But it has its own mitochondrial DNA. And here's a little fun fact about it. Mitochondrial DNA is different from the DNA that you have in your nucleus. The nuclear DNA is a combination of your paternal and your maternal heritage. So half comes from dad, half comes from mom, your genetic parents. Your mitochondrial DNA is only from your mother. So it only comes from the maternal lineage. So if you ever were to test your mitochondrial DNA, it would be 100% identical to the DNA that's in the maternal lineage. So your maternal, or I should say your genetic mother. If you were to test the DNA that's in your nucleus, we would find that half of it would come from the paternal, your father, and the other half would come from the maternal, your genetic mother. There is another organelle that is part of the endosymbiosis theory, so that was free living and was engulfed and made part of a larger cell. We do not have this organelle, but plants do, and that's good old-fashioned chloroplast. So when you look at chloroplast, which plants use for photosynthesis, we also see the fact that it has a double membrane and the fact that it has its own DNA. Here's a look at it. So here you can see the inner and the outer membranes. And spread out throughout the thylakoid membranes, you also see right here the circular pieces of DNA. So the endosymbiosis theory talks about the fact that the mitochondria and the chloroplast were free living cells that were engulfed by a larger cell and made part of their genome. And the pieces of evidence are going to be the fact that it has two membranes and they have their own DNA. But please keep in mind that these organelles still rely on the nuclear DNA to work in a collaborative effort in the cell itself. Now, finishing off the internal workings of a eukaryotic cell, I want to quickly mention the cytoskeleton. The cytoskeleton is what allows the cell to have its shape. If you take a look at this picture right here, you see the little purple lines all over the place. So it will give shape to the cell. It will also allow the cell almost to scaffold, so to move items from side to side or to move organelles around. You basically have three major components of it. You have, starting with the smallest, what we call the microfilaments. Then you have the intermediate filaments. And then last but not least, you have the big ones. These are going to be the microtubules. I just want to quickly just mention the three components because later on we'll talk about how each of them will vary in their overall function. So like for instance, we'll talk about the fact that when you look at your microtubules, they can be used for many different functions. Obviously, structure is one of them. But the cell, if it has a flagella, remember that little whip-like tail, it can go ahead and arrange the microtubules in a more circular order, my apologies, and then will allow for the movement of side to side, which is the whipping action that we see with the flagella. The cells can also use the microtubules for cilia. Cilia is going to be short little hair extensions. For us, we have them in a lot of our orifices, so the openings of our body, like our mouth, our reproductive system, our urinary system. And these little cilia are great for kind of trapping any type of pathogen that might enter into the body. And then your body will wrap it around mucus so it immobilizes it and we can get rid of it. Oh, and then here's a little picture of some sperm cells. Remember, that's the only cell that the human body produces that has a flagella. Now, our last topic for this chapter is going to focus on what they call cell junctions. Cell junctions are meeting points. So we know that we're made out of thousands of millions of cells. How do these cells like interact with each other? Because obviously they need to like connect. Well, it turns out that this is called a cell junction, a meeting point, and there are three options that are very popular. One of them is a tight junction, then you have an anchoring junction and a gap junction. So here on my little illustration, I see that I have four cells that are stacked on top of each other, and as you can see by the plasma membrane, they are connected here by these little purple areas. And these are called cell junctions. And you can have multiple cell junctions within the cells. It all depends on what the overall purpose of the connection is. So for instance, a tight junction means that you're creating an impermeable barrier. So it's almost like you're super gluing or you're stitching the cells together. They are not interacting with each other. They're not sharing any content. They are literally just glued to each other so that they are held together. An anchoring junction is different because it will allow you to not only connect the cells, but it will also allow a little bit of flexibility. So you can see right here, it has a little anchoring junction that allows for the cells to stretch and come back to their original shape. This is what we would see, for instance, in the flexibility of our skin. If you pinch your skin ever so slightly, you notice that it will go up, and if you release it, it'll go back to its original position because it has a little bit of flexibility, and that is all because of the anchoring junction. And the third one is called a gap junction. Now, a gap junction is interesting because when you look at the illustration right here, you're going to notice that the gap junction will allow for the cells to be attached to each other, but it will also allow for a tube or like a pathway, a little channel to form, because the gap junction will have small openings that will allow cytoplasmic content, so stuff from the inside of the cell, to move from one cell to the next cell using the openings in the gap junction. This is especially helpful in, for instance, when we look at our heart cells, because our heart are consistently contracting and relaxing, so they need a lot of electrolytes like sodium, potassium, calcium. So if the cells are able to share their electrolytes, it could be very beneficial to them. And a gap junction will allow the cells not only to be connected, but also to share this internal electrolyte or content that they have. So a tight junction is impermeable. You just kind of glue the cells together. They're not interacting any otherwise. An anchoring junction is good for flexibility, and a gap junction is good for allowing cytoplasmic content inside the cell to go from one cell to the next. Now, keep in mind, these cell junctions allow only for the cell membrane to interact with each other. Some cells have a cell wall. How do we deal with that? Well, if you have a cell wall, what we see happening is that when we look at things like plants and fungi or any organism that has a cell wall, we see that the cell walls will have small little openings or channels that will allow communication between the cells. These are called plasmodesmatas, and if you zoom in here on your stack of plant cells, you can see right here in green is my cell wall, and you can see right here, here's my little channel. Here's the plasmodesmata that allows for the cell to communicate that allows the plant to communicate from one cell to the next, so they can share cytoplasmic content, nutrients, different type of chemicals, and these channels will only appear when you have a cell wall. Okay, and here is a quick reminder: when you look at plant cells, they're mostly made out of cellulose for their cell wall. Bacteria like to use peptidoglycan, and fungi likes to use chitin, which we've mentioned in a previous chapter before. And the last thing I'm going to tell you about this particular chapter is that I know we've listed quite a few organelles. There is this really nice table in the book, table 3.2, that will allow you to see a list of all the organelles we have discussed. It will talk a little bit about the structure of the organelle. More importantly, it will talk about the function of the organelle. And in the last two columns, it will give you a little reminder as to which one you will find in both animal and plant cells, or exclusively in plant cells. And remember, we talked about the fact that in plant cells, they will have a vacuole that we don't have, and they will, of course, have chloroplast, which we don't have, and a cell wall. I forgot about the cell wall. All right, everyone. I hope you enjoyed this chapter. And as just a reminder, any questions, comments, or concern, please do not hesitate to reach out. And we shall talk again soon. Bye.